3, the grades ranged from 97 to 2. You now have all the data that you need to determine your class standing. The only thing left to do is the homework number 13, but you can calculate your homework average from the grades that you have, and it should come out pretty close. The reason you want to know your standing is so that you can make a decision which final exam to take. And so remember, if you need to drop an exam, then you can take the comprehensive final. What the comprehensive final looks like is 2 exams stapled together. A standard 1-hour Section 4 exam based on this last week and a half's worth of work, stapled to a standard 3-page review exam based on questions drawn from the first, second, and third sections. Those questions on the review part of the exam can either be stuff that you studied for and I didn't ask the first time around, or it can be questions that lots of students missed and I ask it again. Like I've said a number of times before, don't try to guess what I'm going to ask. Too many people did that and lost lots of points because they didn't know one of the genetic systems. They just assumed it wasn't going to be on there. You need to know everything. You can't guess what I'm going to ask. When you pick up your exam, exam 3, go through it carefully. If I couldn't figure out what you were trying to say, if I couldn't read your handwriting, I just took off all the points. You can come in and read the answer to me. I've given back a few points here and there so far. What you want to be sure of is if you're taking the comprehensive final, that you understand the questions that you missed, actually on all 3 exams, actually on the in-class cooperative learning exercises, and the homeworks. You probably noticed that homework questions occasionally pop up on the exams. So now you have to figure out which exam you're going to take. Here's the way it's going to work. You do not need to make that decision until Monday when you walk in this room. Actually, you don't even have to make your mind up when you walk in the room. But as you walk down the aisle to this table, there will be 2 stacks of exams. That's when you have to make up your mind. Actually, that's not true either. You can take the comprehensive final exam and you can look through it and you can decide at the beginning or halfway through or at the end if you don't want to take the comprehensive exam. I'm very serious. Halfway through the final exam period, the TAs will say, anybody taking the mini-final, turn your exam in now. There will then be a sealed pile of mini-exams. Anybody who does not turn the mini-final in halfway through is automatically going to be graded on 200 points for the comprehensive final. So that's the way it works. I think you would be better off making up your mind really pretty soon so you know which material to study. Remember, if you don't need to take the comprehensive exam, then you're only studying a week and a half's worth of material. Otherwise, you really have to go back and review all the material if you want to drop one of your exams. Now here's one other thing that I'm going to do. For everybody who takes the comprehensive 200-point exam, if you pass that exam, no matter what your average is, if you pass with 65%, that's 130 points, you will automatically pass the course. If you get an A on the comprehensive exam, that's 85%, 170 points. If you get an A on the comprehensive exam, no matter what your average, you get an A for the course. That means that for everybody who takes the comprehensive exam, I will go through your grades and look at your final average and your exam score and give you automatically the highest of the two for your final grade. Every year, every semester, there's always a few students who jump a grade, who end up with a grade higher than their average. But you really have to know the stuff. I mean, that really works for a student who's been working all year, struggling, and then just put it all together at the very end. And so that's what's on the table. The exam is a week from Monday. I'll post everything up on Monday. Everything will be the same except there's no review sessions. I'll be around the entire time of final exams and that intercession, that Wednesday in between the end of class and exams, stopping any time with any questions. The exam's a 2-hour exam, and we'll be here. I'll post the information up. I will put the old exam, last year's final exam, up in the copy center. No surprises. I mean, you know by now, you have 6 of my exams. You know the kinds of questions I ask. The format will be exactly the same. Anybody have a question? Yeah.
Where do we find like range of grades, like 85 to 100 exams?
Well, that's what I told you on day 1 when we went over the syllabus. The cutoffs are 85, 75, 65.
When we answer for the test, can you just like take one of each and then practice the table on the students?
No, you don't need one of each because the comprehensive final has one of each. So if you're undecided when you walk in, take the comprehensive final, sit down, look at it. Don't spend too much time. I mean, it's still a timed exam. No, next Monday is the last day of class. It's a week from Monday. It's the final exam that's in the final exam schedule that ASU has up on the web on a Monday, Wednesday, Friday class at whatever we did noon.
Will there be a review guide like you put in the copy center for the previous exams?
Yes, I will put last year's final exam in the copy center.
Would that be the mini or the comprehensive?
No, the comprehensive.
Okay.
Full exam. So you see everything and what it looks like. I mean, again, the structure is good. 2 exams stapled back to back.
Will that be up on like Monday or Tuesday?
I don't know.
All right.
I haven't thought about it. It's too far in advance. I'll put all that information up next Monday, this coming Monday, and tell you exactly when it will go up. Someone had a hand up there? Any last questions on how the end of the semester will play out? You have all of your homeworks. Only thing that's left is homework 13, and there are still lots of exam 1s and exam 2s that haven't been picked up. If you're interested, you can stop by my office and get those also. Any last questions? Yes?
So there's not a homework 14?
No, the one you have now is the last one. It's due Monday.
And we dropped 2?
You drop, that's right, you drop 2 homeworks. Your 2 low homeworks will automatically be dropped. PAs will take care of all that for you. Anybody else? Okay, so we've been talking about selection. Yes. It's 10 after 12. I have no proof. Post it on the web. Any other questions? So we just spent a fair amount of time going over selection. We had a whole chapter on mutation. We looked very briefly at how mutation and how selection affects population structure. And so the big picture, you know the parts of the big picture, now we can put them together. The big picture is, as long as you are alive, mutations happen. Mutation is a natural process. You cannot stop it. It's a consequence of having ring structures for your bases in your DNA. And so mutations happen all the time. As mutations come into the population, those mutations are evaluated by selection. So there is a very tight correlation between mutations that enter the population and selection that judges those mutations at that time. Most mutations are neutral. Literally hundreds of mutations, every person, every generation. Most mutations have no effect on the phenotype. Some very, very few are bad and are selected against, but some very, very few are good and are selected for. We only look at one small component, and that is how selection against a recessive allele works. There are all kinds of other formulas to do all kinds of other things. We just look at a part of that. So the bottom line is that mutation and selection are inexorably intertwined for individuals. Selection is at the individual because of fitness, how many progeny that individual leaves. That's all that matters. Doesn't matter how smart they are, doesn't matter how rich the person is. All that matters is how many progeny are left. So there's a very simple formula to connect those two. Q at equilibrium, that's all there is. The mutation rate is fairly easy to calculate for almost all genes. For the most part, it is simply based on the size of the gene. We went through this, I don't know, weeks ago when we talked about mutation. It doesn't show up very well. Ooh, that's a good one. Selection we've already talked about. Again, selection at an individual. Selection can change over time. As the environment changes, selection can change. If we're in a wet period, then mutations which confer drought resistance are not going to be selected for. They might even be selected against. As the environment changes, ice age, global warming, drought, all of those things affect the global set of alleles in an individual. And so this is not a forever formula. Selection has to be determined in every environment. Any questions about what that means? We're not going to do anything with it.
Is that mu for mutation?
Mu for the mutation rate, yes.
So mutation rate over selection rate?
Mutation rate over the selection coefficient.
Okay.
Remember, that's the inverse of fitness. Anybody else? All right. So what we did on whatever it was, Monday, I guess, looking at selection, what we saw is that even if we have absolute selection, zero fitness for an individual carrying this mutation, that mutation is still around the next generation. We talked a little bit about that, and you understand that the reason zero fitness doesn't eliminate a really bad mutation, there are two reasons. First of all, those really bad mutations are hidden. We did recessive alleles. They're hidden in the heterozygote. And so they're completely impervious to selection. Not all mutations are, but most recessive mutations are. Otherwise, we call them semi-dominant or codominant, something like that. The second reason that the mutation will never disappear, even a dominant mutation, is recurrent mutation. And so every generation that you eliminate alleles because that individual doesn't leave enough progeny, or maybe doesn't leave any progeny, is because every generation, the mutation is going to put more mutations into the population. And so we always have that rare allele, all the human disease mutations, those are all rare alleles. There's a second way, I guess third way, in which mutations can persist in a population, and that is called overdominance. Also called heterozygote advantage. So let's take two situations. One is hemophilia. Hemophilia is a pretty bad mutation. It's near lethal. And so the wild type has a fitness W of 1.0, a heterozygote 1.0, and the homozygous recessive probably about 0.1. I'm just guessing, but it's pretty bad. Well, hemophilia is one. The second one is sickle cell anemia. That's an interesting mutation. It's also a blood mutation. That is a mutation of the red blood cells, a mutation of the globin genes that we looked at. What sickle cell anemia is a single base pair change that changes one amino acid in one of the globin chains, I don't remember which one, that changes the shape of the hemoglobin protein, which changes the shape of the cell. So it's called sickle cell because as you look at it in a microscope, the normal red blood cell looks like a hockey puck, a dimpled hockey puck. Sickle cell anemia, the red blood cell is curved like a dish almost. And that makes it very difficult to flow through the tiniest arteries and veins, and they tend to get clogged, and it's a pretty serious disease. But way back in the teens or 20s, as Mendelian genetics popularity grew, people were doing all kinds of genetic studies, including population. Remember, Hardy Weinberg came really early. What people discovered was that in certain regions of the world, there was a discrepancy with Hardy Weinberg. What you expect in Hardy Weinberg, they weren't finding. What they were finding was a fitness for the wild type of 1.0 for the heterozygote 1.2, and for a really serious disease, a surprising high allele frequency in the homozygous recessive.
Isn't the sickle cell anemia the reason why the heterozygote is advantageous? Because in certain parts of the world where malaria is prevalent, it gives you an advantage against like the malaria.
Did you hear what he said? He just stole my whole thing. That's exactly what's going on.
Did no one not,
The
did no
reason
one not know that?
for this,
You didn't know that?
No, I knew that.
we
Okay.
have a high number for the homozygous recessive, higher than expected, is because the mutation called sickle cell anemia in the heterozygote, that mutation doesn't allow the malaria to replicate properly, And so the heterozygotes are protected against malaria. And so this phenomenon was found all around the Mediterranean. We're talking 20s and 30s before they were spraying with DDT to eliminate them. And so overdominance means that because this really detrimental allele, little a, is really bad for the homozygous, it's beneficial for the heterozygote. Hence, heterozygote advantage for overdominance. Dominance should be 1.0, this is 1.2. There are actually 2 or 3 other genetic mutations that have exactly the same effect for exactly the same reason. So that's another reason why a really deleterious mutation, little a, is overrepresented in the population. Any questions on that? Yes?
So it has more in the homozygous recessive because the heterozygous have more?
Yes, because, that's right. That the allele frequency here is higher, that means that these are going to make more homozygous recessive. That's exactly right. Yes?
I know it's not the same thing, but because it's a fitness effect, but if you have, if there's selection for vigor and you have hybrid vigor heterosis, would that affect this as well? Even though it's slightly different phenomenon? So it's
I don't have an answer.
Yeah.
I don't know the answer to the question. Any questions on overdominance? So remember, the Hardy-Weinberg law says that as long as you know the allele frequency, P and Q, you can calculate the population structure with the binomial, P squared plus 2PQ plus Q squared. So that says with a given allele frequency, the population structure is absolutely locked in. It does not change. But we all know that the population changes. The population changes because of migration, because of mutation, because of selection. Those are considered as pressures. That means that if we know the force that's happening, if we know the direction of migration, if we know the mutation frequency, if we know the fitness, you can calculate exactly how that population is going to change because we know how the allele frequencies will change. The fourth is different. Drift, also called genetic drift, is not a pressure. It's not a force. So it doesn't work the same way. Oh, and by the way, we're going to come back and talk about migration soon also. There's some special issues with migration that are going to be very important for the next chapter. So right now we're looking at genetic drift. Genetic drift, very simply, is random sampling of a gene pool. So I always say gene pool, but we know it's not genes that are being, it's alleles. And so that's a bad habit that I've gotten into. And you should know the distinction between the two. It's random sampling of the alleles at every generation. So I'm going to give you 2 examples. One for a rare allele, and then we'll look at what happens with common alleles. Because drift happens with every allele at every every single generation. So let's start off with 500 individuals all having homozygous big A. These 500 individuals are in a stable population. That means that each couple, 2 people, have 2 children on average. So now let's assume one mutation in that population of little a. That makes 999 big A alleles and 1 little a allele. The upshot of that is, after that one mutation to little a happens, everybody is mating, everybody's having two children. We have mostly big A, don't care. We have one couple that's homozygous big A, one individual in the couple, and one individual that's heterozygous. So what that means they should have one of each. Punnett square. But Punnett square is not a guarantee of what's going to happen. They could end up having 2 big A's. Or they could end up having 2 heterozygous. Or one of each. If both of their children are big A, that new mutation disappears. It's gone out of the population. You cannot resurrect it. Or, equally probable, they can have 2 heterozygotes. That means in one generation, the allele frequency has doubled. That's why I said that drift, genetic drift, random sampling, you cannot predict the direction of change of allele frequency. With a mutation, you can predict big A to little a, or the reverse. With selection, you can predict if little a has a very low fitness, that it's going to decrease. With migration, you can predict if dark-haired Italians move to Sweden, that the blonde allele will decrease, dark hair allele will increase. But with drift, you absolutely have no way to predict. Does everybody see what's happening here? You see why I say it's random sampling? Let me read a statistic to you. The risk of loss is so great for rare alleles that only one mutation in 10 that is neutral, no selection, will be present after 15 generations. What that means is even if we got that or that, the next generation, the same situation is going to occur. In the next generation, it can either disappear, stay the same, or double. Every generation, once it's gone, it's gone. Let me finish that. Even even if the allele is beneficial, even if it's the greatest allele in the world, allows you to shoot baskets with 100% efficiency, take exams and score 100% every time, even if the allele was the greatest in the world, can't do a thing about it. Absolutely nothing can be done to override this genetic fact of random probability. That's what random sampling means.
So you said that the mutation will never disappear, but if it disappears right there, are you just saying that it could come again?
No, I didn't say it would never disappear.
I thought you said a mutation will never disappear because
The
it'll be
risk
hidden
of
in
loss is so great for rare alleles that only 1 in 10 mutation that confer no selection will be present after 15 generations.
But earlier we said because it's hidden in the heterozygote.
Oh, we're talking about a large population
Oh, okay.
with multiple alleles. So that's already gained a foothold. That's completely different. That situation up here is completely different.
What was the 1 in 10, is it 1 in 10 will be present after 15 years, or no one will be present?
No, only 1 mutation in 10 will be present after 15 generations. And just making it a good allele, changing it from neutral to good, has no effect, because none of this is about selection. It's all about random probabilities. If this individual is out for a walk and gets jumped by a lion, that allele's gone. Once it's gone, it's gone. Random sampling. Nothing can be done to alter the statistics on this system.
So if we were trying to think about it, it would just be, if we were just trying to think about it like simply, it would just be the probability that a new mutation stays.
I didn't hear the beginnings.
To think of it simply.
So random drift, genetic drift is random sampling of the gene pool. Once an allele happens, then it's subjected to just natural phenomena, including getting eaten by a lion, including that as a significant possibility in passing that allele on. You cannot alter that, even if it's a great mutation. Anybody else? Okay, so this is all about a rare allele. Now let's look at a common allele. Because drift happens to every single allele on every single chromosome. So let's start off with a population. And we're going to do we're going to just measure, we're going to set this population up in a cage so we can control the initial parameters. 100% big A, 100% little a. So we're starting off with a population that is 50% big A, 50% little a. And then we're going to let this population go, but we're going to restrict it such that we have a stable population size. And so every generation, we're just going to randomly cull the population down so we keep it at whatever you want, 500 or 1,000 individuals. So you don't even have to be able to see the allele, but you can test for it. And so what you find invariably is random fluctuations. That's one scenario. time for, I don't know how many, countless number of different species that are examined, a common allele in a small population will always end up the allele will get fixed. That's the terminology. When the allele is fixed, it says one or the other. Toss a coin, absolutely random. One or the other allele disappears, and the other allele will become 100%. This is all about random sampling. It happens no matter what. This is population size dependent. That means that the larger the population, the slower is fixation. And there comes a point, I don't know what that point is, but there is a point where the population is large enough that introduction of new alleles by mutation balances loss of alleles by drift. The real consequence of drift is that populations of endangered species are at extraordinarily high risk of losing genetic diversity. And so think of any population, condors, cheetahs, whatever it is, small populations run this risk at a much greater level than a large population. It's impossible to happen in humans, 6 or 7 billion individuals. But there are lots of populations of plant and animal species where the loss of alleles due to drift is much greater than the mutation rate to replace them. That's the tipping point. We can replace our alleles because we have 6 billion people. Someone ever hear that? Oh, just 3 random populations. There's no one pattern. I mean, it's just all over the place until it hits one or the other axis. Yes, so the green one is going to fix homozygous big A, the black and the blue is going to fix homozygous little a.
You said endangered species would lose what kind of diversity, genetic diversity?
Right, endangered species have very low genetic diversity, which means they are at very high risk of losing rare alleles.
Is there a higher frequency of fixing on the dominant allele than the recessive?
As long as there's no selection in play, no. Even if there's selection in play, remember I said even if selection is in play here, that doesn't change the scenario. It may modify it slightly, but it's not going to alter the final outcome.
Would you define fixation as just when one of the alleles is brought back to population?
Yes, or the other allele went to homozygosity. Either way is fine. Any questions on random drift? The bottom line is random drift happens in every single individual every time an individual is born. There is no guarantee of getting the alleles you want. There is no way around random drift. It doesn't matter how large or how small the population is. Alleles will drift out of the population randomly. Any last questions before we move on? Yes?
So, just to try to get this right, every allele of every generation is subject to drift?
Every allele in every generation is subject to random drift. That's exactly right. Anybody else?
So how does it also apply to great? Yep. So like in the animal kingdom, lions, for example, lions are bigger than me, but some of them shows off more like how does genetic drift? You have selection.
Because those are really small populations, and they're still losing genetic diversity. Lions aren't yet endangered. I just saw something today that population in Africa has dropped from 100,000, I don't know, 10 years, 20 years ago, down to 30,000. All species take the incredible toll. Any last questions? Okay, so the next chapter. Next chapter is typically evolution, but we're going to couple together origin of life and evolution as just an extension of what we have done. And so, evolution is a scary term for some people. Evolution has a very simple meaning. It's just change in a real frequency. That's all it is. There is no other definition. Systems evolve, that means change, and that change happens in a real frequency. That's why in virtually every textbook, chapter on evolution follows the chapter on what we just covered in population structure. And so we just went through all those forces that change the allele frequency in a population. But I'm going to start this chapter off a little bit differently because evolution needs something to work on. And that something is origin of life. And so we're going to take a kind of a close look at how life began on Earth to allow evolution to work on it. So first of all, some numbers. The Earth formed about 4.5 billion years ago. That's a fairly accurate number, but I'm not going to bother with the plus or minuses. That's not all that important, whether it's 4.5 or 4.6. That's when life originated. Out of a cloud of planetismals, meteors, all sorts of debris that coalesced into the planets circling the sun. And for the next millions of years, hundreds of millions of years, Earth was bombarded by meteor showers, and life simply wasn't possible. Earth had to cool down, and so first life appeared at about 4.2 billion years ago. Earth became habitable, Earth became habitable at 4.2. No, 3.85, that's the one. So what we have here is a window. So the geologists say that the Earth had cooled off sufficiently. Earth got hit by a giant meteor that spun off the moon, which is an interesting story in itself. But by about 4.2 billion years, the Earth had stabilized. The first evidence for life is at about 3.85 years. Now I say evidence of life because we're not talking about of bones that leave skeletons behind. We're talking about evidence that organisms, microorganisms, bacteria, have changed the environment. And so the evidence is really getting very good at pinpointing down when life originated. And that was about 3.85 billion years ago. So microorganisms. Eukaryotes didn't come on the scene for several billion years after that. So first life existed for a long time on Earth. And so the question is, what is the origin of life? The short answer is, I have no clue. All kinds of models exist for how life came onto Earth. The least palatable is that a spaceship arrived on Earth and deposited life. But I don't like that because that just puts off the argument one planet further. Okay, so what about my planet? So, bottom line is, we do not know how life originated. But we do know what the conditions were in the window. And the conditions in this window of opportunity, a long time, hundreds of millions of years. In that window, Earth looked and was nothing like today. We live in a rusted out world. There was virtually no oxygen on Earth until green plants came along billions of years after life began. And so we're in an oxidized world. Life did not begin under these conditions. Life began in a reducing atmosphere with a totally different mixture of gases. And so you cannot compare life today with what happened in that window. We really have little or no idea of the sequence of events that led to the first organism. But we have some constraints that are really interesting that we can put on that first organism and those first organisms. So first of all, we have this idea of ribosomes. Talked about that. All your critical covalent bonds, they are not made by protein enzymes, they're made by ribosomes. That is, the ribosomal RNA makes your peptide bonds, no protein enzyme involved. Ribosomes also can make and break phosphodiester bonds. We don't do much of that now, but the splicing is by ribosomes. So this whole fantasy about which came first, proteins or nucleic acids, has no meaning. And from this notion of ribosomes, we derive the idea of an RNA world. People who do this kind of work have this notion that the first living organisms, by any definition you wish for a living organism, the first living organisms did not have DNA, but they were RNA based. In fact, most people now believe that even the RNA world, that something preceded the RNA world. The RNA world then gave rise to our DNA world. The RNA world is pretty firm. know we do not need proteins to undertake complex chemical reactions. But RNAs to do those reactions. Organic compounds is actually a misnomer. The original idea of an organic compound is a chemical made by living organisms. The original derivation of the word. But we now know that many, but not all organic compounds, can be made non-biologically. For example, there are amino acids on meteors. Amino acids formed in outer space. Non-biological process. And so it is very easy to imagine an accumulation of what we call organic compounds non-biologically. Making the jump from a collection of organic compounds to a living cell is still something that's very hard to imagine. But there are some constraints that are really very important. So, first of all organisms on Earth today have the same genetic code. That's really important. That's an important clue. Now remember when we did the code, I said a universal code almost. The almost does not include anything that's very different. It includes organisms that have slight variations, offshoots of the standard code. That includes mitochondria and chloroplasts. So every living organism has the same genetic code, basically. That puts some constraints on what that first living organism was and how it developed. What that says, only three possibilities, is life originated only once. That's one possibility. And from that one-time, super rare event, all life has descended. Or life multiple times, identical code. Probability of that is like 0.0000000. That something that complex could have happened numerous times with identical genetic code is beyond the pale. But there is another more reasonable possibility, and life multiple times, different code, only one survived. And that's a real distinct possibility in my mind. Because the numbers say that something like 99.99% of all species that ever inhabited this planet have gone extinct. What's left right now is really just the tip of the iceberg. That most, the vast majority of species have disappeared. And we have absolutely no clue what the genetic code of those species could possibly have been. And so, one or three are in fact possibilities. But we don't know. There are actually things that we do not know. Any questions on that? Yeah.
For the 4.2 billion years ago?
For what?
4.2 billion years ago, did we say life, first life form, or it was inhabited?
No, 4.2 was when the world became inhabitable. And so, 300 million years of bombardment down to 4.2, and it took about 400 million years. No fossil evidence, understand, no bones, but evidence of biological enzymatic activity altering the environment. That's what it's all about. Any questions on that? Okay, so everybody agrees that life has existed on Earth for a long time. As long as life exists on Earth, because of that top board, population structures change over time. Ice age causes significant change in population structure. Drought causes significant change in population structure. Heat wave causes significant change in population structure. That's all evolution is change in population structure. So, since selection is the key, selection is fed by mutation, what does selection have to operate? First of all, mutation feeds into selection. But anything that alters population structure, and again, by that I mean p squared plus 2p2 plus q squared, real frequency. Anything that feeds into that, including Mendel's independent assortment and recombination happens automatically and all the time. So what is population structure? What does population look like? First of all, population these are the critical ones. We'll talk about migration. First of all, population has high heterozygosity. That means that as we look in the population, heterozygous genes are pretty much the rule. Populations have high polymorphism. So heterozygosity is at the individual level. Probably somewhere around 40 to 50%. Each one of your genes is heterozygous. So a very, very high level of heterozygosity. Very high variability. Polymorphism is population. It means multiple form. The most common allele is less than 95%. So hair color is not, or is, a polymorphic allele. Eye color is a polymorphic allele, gene. But BRAC1, which has dozens of different alleles, BRAC1 is not polymorphic because the most common allele, wild type, is greater than 95%. So there's high polymorphism, high heterozygosity. Together with Mendelian genetics, independent assortment and recombination lead to a tremendous diversity. We've only within the last few years gotten a really good look at what individuals look like at individual DNA level. And so as the cost of DNA sequencing has come down dramatically, somewhere between 1,000 and 2,000 people, maybe even more at this time, have had their complete genomes sequenced. The first genome sequencing cost $1 billion. $1 billion to get the first human sequence. And that was pooled from multiple individuals. Now, individuals can get their entire genome sequenced for probably just under $10,000. And the price is coming down rapidly, almost the way memory chips are expanding exponentially. And so, only within the last few years, people have sequenced entire family, mother, father, and children. So able to ask the question, exactly what is inherited? What kinds of mutations? Let me give you some numbers. An individual has SNPs, which stand for single nucleotide polymorphisms. You have 100,000 or more SNPs. That is a nucleotide, a base on your DNA that's different from most people. And we have 3 billion bases. So we have SNPs. We have CNVs, copy number variants. CNVs are very common. We've talked about satellite DNAs, but that's not it. We're talking about the microsatellites. Thousands of them scattered throughout your body. That's what DNA forensics looks at, is copy number variants. And those can expand. Huntington's and Fragile X disease. We have indels, turn out to be very common. Indel is insertion, deletion. Your genome is just riddled with thousands of copy number variants. At a less level, we have translocations and inversions. We don't have a lot of those. Other species like Drosophila seems to have way more than we do. We have magnifications that lead to Huntington's and Fragile X. Duplications really are special. You have all sorts of duplications, ranging from small duplications to large duplications that happen all the time. That's the only way that species have become more complex. So first of all, we have small duplications. If you look at the immunoglobulin genes, what you see is the light chain and heavy chain are related. Heavy chain is just a set of duplications. The globin genes are a set of duplications. Only by duplicating the globin genes and having embryonic, fetal, and adult is placental life possible. As life becomes more complex, organisms need more genes. Once we have a duplication, Then we have mutation and divergence. Silk is a gene with internal duplications. So an original globin gene duplicated a long time ago. And we went from an adult copy to an adult and a fetal copy. But once we have two copies, one of those copies can mutate freely, take on new functions. Duplication and divergence gives rise to new functions. Just look at all the tRNAs. Look at the globin genes. Look at the immunoglobulin genes and gene cassettes. And so that's a powerful way to get more genes. All higher organisms arose because of whole genome duplications. Humans have countless number of whole genome duplications in our evolutionary history. Even an organism as simple as yeast, the yeast that you played with a few times during the semester, that simple yeast is the result of a whole genome duplication. You have 4 sets of homeotic genes. Drosophila has 1 set. You're more complex. You need more diversity. And so duplications are a way for life, for organisms, to become more complex over time. Any questions on what does this population look like? Yes?
What are CMVs again?
Copy number variation. So your microsatellites, you may have 6 copies of satellite 7, I'll have 7 copies. And that's what forensics looks at. That's why it's so powerful, is because there's no selection for or against it. And so they're just random variations in number. Yes?
So is this things that are different between like a species and itself?
That's the difference between you and me. So this is what distinguishes individuals. We differ because of single nucleotide polymorphism. Because you and I have, for sequence X, you may have 10 copies, I may have 15. Insertion, your genome is just riddled with small insertions and deletions. Remember, only 2% of your DNA codes for protein. 98% does not code for protein. So we can have lots of mutations in between genes. Translocation and translocation and inversions, not so common in humans, especially inversions. We talked about that way back when we covered that chapter. But duplications, that's critical for a species becoming more complex. How do species become different? Species become different by regulatory mutations. Mutations in the homeotic genes. You all know what a millipede or centipede is, right?
Yeah.
So that's an insect like Drosophila. The antennapedia mutation that we talked about very briefly is a mutation that suppresses leg formation. A millipede and centipede have a leg on every single segment except the head. Insects, true insects, like flies, only have legs on the thorax, suppressed in the abdomen, and suppressed in the head segments, unless you mutate the antennapedia gene, which then allows the leg to grow out. And so homeotic gene mutations is what gives the ability for organisms to become really different. Some insects have one pair of wings, two wings, like flies. Other insects like dragonflies, you know what a dragonfly is? Two pair of wings. Dragonfly has wings on two thorax segments. Drosophila has wings on one thorax segment. Those kinds of changes that sound really difficult, one simple mutation can go from a four-winged dragonfly to a two-winged fly. Any questions? What makes everybody different? Any questions on that? When we come back on Friday, we'll finish up this chapter.